Hey guys, welcome to Bio 112 Online, your home for biology education. We have come to the final topic of the semester, osmoregulation. So for osmoregulation, what we mean is the process of maintaining homeostasis by regulating solute, concentration, and water balance. So what do we mean by osmoregulation and osmolarity? Osmolarity is going to be the level of osmotic pressure or the total solute concentration per liter of water. This goes along very closely with other terms we've used this semester, specifically when we've talked about things like tonicity. In the case of tonicity, we're talking about the salt concentration outside of a cell relative to the salt concentration in the cell. In the case of osmolarity, what we're simply saying is we're measuring the two salt concentrations on either side of a membrane, regardless of which side has cells on it or not. Osmolarity in that case is going to be a comparison of solute concentrations between two solutions. If those two solutions have the same solute concentration, we would refer to those as isoosmotic. If two solutions have differing solute concentrations, the solution with the higher salt or solute concentration will be referred to as hyperosmotic. The other solution of the pair, the one with less solutes, would be referred to as hypoosmotic. Now we refer to these because they have the same general principle and the same general result as tonicity. If you have isoosmotic solutions, then there will be no net movement of water between the two. If you have a difference and one solution is hypoosmotic and the other is hyperosmotic, water will always flow from the hypoosmotic solution to the hyperosmotic solution. We can bring this concept now to the point of animals. And we've already kind of touched on this when we were talking about the formation of homeostasis and the ability to adapt to the external environment. We said some organisms are going to be conformers. They're going to maintain their internal conditions the same as what's on the outside. Regulators, on the other hand, were organisms, or in this case animals, that will have a differing internal environment relative to what's outside the environment. This brings us to our discussion of regulators and osmoregulators. An osmoregulator is going to be an animal that controls its internal osmolarity independent of whatever's going on in the external condition. A good example of this is fish. Now, you normally think of fish swimming out in water. Well, the thing is that the fish's internal bodily fluids are going to have a higher salt concentration than the water surrounding them within freshwater environments. This means that the water outside is This means that the water outside of their body is hypoosmolar relative to the solutions inside, and water will have a tendency to want to enter. The fish deals with this in one of two ways. The first of which is it will begin to collect salts through its gills, and the second thing that it will do is use its kidneys to secrete a very dilute urine to get rid of excess water. In the case of saltwater fish, they have the opposite problem. Their internal bodily solutions are going to be hypoosmolar relative to what's outside. As a result, they're constantly losing water to their surroundings. The fish gets around this by drinking lots of water, salt water, into their body, and then excreting the salt through their gills. So we have two things that are going on here. One of which is we're controlling how much water we're bringing in, and second off, we're controlling how much salt is entering and leaving the body. This is going to be critical for our next understanding. In the case of terrestrial organisms, we have essentially the same problem, but we have one further step to it. In the case of terrestrial organisms, we're not surrounded by water, we're surrounded by air. As a result, we're constantly losing moisture to that air. We have to replenish that. At the same time, we have an internal salt concentration, and we have to match that internal salt concentration with the set point at all times. If things get too salty, then that starts causing problems. As a result, we have to manage both the amount of salt and water in solution within our bodily fluids, matching the losses and the gains. In the case of terrestrial animals, the way in which they're gaining water is, well, they drink water, and also they get water from their food. Recall when we were talking about cellular respiration, one of the products of cellular respiration was water. In addition, terrestrial animals will go through and try and reduce the amount of water that they're losing by having some type of cuticle or surface, i.e. our skin, the exoskeletons of insects, that kind of thing, to prevent water from evaporating off. Finally, the amount of water that can be brought in attempts to be matched between what is evaporated and what is lost via the process of excretion. Excretion is going to be the process of disposal of nitrogen-containing metabolic wastes or other wastes from the body. The body builds up lots of toxins, and then we have to flush some of that out. That occurs through the process of excretion, and inherently has some level of water loss associated with it. When we talk about nitrogen-containing waste, what we're talking about is metabolic waste that are generated especially from the breakdown of proteins and nucleic acids. Recall when we were discussing digestion, we said that you can break down proteins into amino acids. And we said that those amino acids can then, at times of need, be fed into varying aspects of cellular respiration, either converted into pyruvate, plugged directly into the Krebs cycle. But in order to make that happen, the first thing you have to do is take off that nitrogen, that amino group, from the amino acid. That amino group, once it's been cleaved off, becomes an ammonia. This can become problematic if it's happening too much because ammonia is toxic if it gets too high of concentrations. In the case of fish and amphibians, which spend their time in and around the water, what they'll simply do is excrete this as a dilute solution straight out into the water. In the case of fish even, what they'll do is they'll use their gills and simply excrete the ammonia through their gills. Now we said that terrestrial animals, especially the tetrapods, run into other problems with this. Particularly in the case of mammals, we can't lose so much water. As a result, we have a tendency to take that ammonia and convert it into a less toxic form called urea. In the case of birds, they have a second problem entirely. That is that they like to fly. As a result, they don't want to have a whole lot of water stored in their bodies sitting around just to be used to flush out wastes. And so they hyperconcentrate that nitrogenous waste into a product called uric acid. In that case, during the disposal of that uric acid, they only can excrete a very small amount of water, creating lots of water retention ability, meaning they don't have to drink so much and carry around so much. In order to go through and do this process, whether we're talking about amphibians or reptiles or birds or mammals, we all have to have some type of excretory system. This excretory system will regulate the solute movements between internal fluids and the external environment and has 4 generalized stages. These 4 generalized stages are referred to as first, filtration, second, resorption, third, secretion, and finally, excretion. Filtration is going to the process of the extraction of water and small solutes from the bodily fluid. It is going to be, as the word suggests, to filter out the chunky bits. This is going to take something like a solution of blood and remove the blood cells and remove the large proteins and allow the smaller molecules to pass through. These smaller molecules, once they've passed through the filter, are referred to as the filtrate. That filtrate will then undergo the next process referred to as resorption. This happens because many of the small molecules are things that we might indeed want to keep. As a result, the process of resorption will be the process that retains valuable substances returned via active transport across some type of transport epithelium from the filtrate to the body fluid, or taken in a much more simple phrase, to return the things that we need back to the body fluids. The flip side of this is the process of secretion. The process of secretion is preferentially taking things from solution in the body fluids and actively transporting those into the filtrate, which means you're losing them at a much higher proportion than they existed previously in the body fluids. Therefore, secretion becomes the removal of toxins or other unwanted solutes by actively transporting them from the body fluid to the filtrate. The fourth step in this, after you've filtered out the chunky bits, after you've resorbed the things you want, after you secreted the things that you don't want, is finally the process of excretion, or the process by which those filtrates leave the body. When we were discussing this, in both the cases of secretion and resorption, we're relying on transport epithelia to conduct those processes. Transport epithelia are going to be specialized epithelial cells that regulate solute concentration within the interstitial fluid and are responsible for moving solutes and water from one side of a membrane to the other. By membrane in this, I'm referring in both cases to the membrane of a cell and the membrane formed by a group of cells. In both of these cases, they're playing essentially the same roles. This is going to be due to a complex set of tubes with a relatively large surface area. In this case, it's going to be epithelial cells with a brush border or microvilli. In this case, the solutes that are being moved by active transport, which means that there's a certain level of specificity to the movements. Specific solutes are being transported in a specific direction. So for example, if you wanted to move salts from one side of a membrane to the other, the first thing you're going to need is sodium-potassium pumps. This will draw down the sodium concentration inside the cell. The next thing that you're going to need is going to be sodium channels that will allow sodium to pass from one side of the membrane into the cell, which would then be pumped into the solution outside on the basal end of that epithelial membrane. As a result, you have a net movement of ions from one solution to the next. This is going to be a critical component of the process of the kidney. The kidney is going to be an organ responsible for regulating osmolarity and removal of nitrogenous wastes in many vertebrates. This is going to include the tetrapods, such as birds, mammals, amphibians, and reptiles. The kidneys are going to be two roughly fist-shaped organs located right here above the small of your back. Blood flow into those kidneys is going to be regulated by the renal artery, which is going to branch off from the aorta and allow blood to flow into those kidneys. Returning from the kidneys will be the renal veins. Those renal veins will flow then directly into the inferior vena cava. The filtrate that's produced within the kidneys will be converted and processed into what we refer to as urine. That urine will then exit the kidney via a tube referred to as the ureter and will be transported to the urinary bladder. Once in the urinary bladder, that urine will be stored for some period of time before it is eventually emptied out via the urethra to the external environment. In the case of the mammalian kidney, it's going to be composed of three main zones. The first of these, the outermost layer, is referred to as the cortex. You'll be familiar with this term when we're doing our discussion of the adrenal gland. The outer layer is called the cortex. The inner layer of the mammalian kidney is referred to as the renal medulla. Again, similar to our discussions of the adrenal gland, where the inner layer was the medulla. The innermost layer of the kidney is referred to as the renal pelvis. This is going to be an area where all the collecting ducts come together, and we'll connect all those collecting ducts to the ureter. The functional component of the kidney is referred to as the nephron. The nephron is going to be a relatively long tube composed of transport epithelia that is going to be responsible for osmoregulation in the kidney. It's going to have the four main processes going on. It's going to have the process of filtration, of resorption, of secretion, and excretion, all taking place within this nephron. That filtrate of the nephron is going to be processed as it moves through four main zones. Those four main zones are referred to as Bowman's capsule, the proximal tube, otherwise known as the proximal convoluted tubule, the loop of Henle, and finally the distal convoluted tubule or the distal tubule. As the filtrate passes through these series of zones or regions of the kidney, you will have different processes taking place. The nephron will connect to what's referred to as a collecting duct, along with several other nephrons. Those nephrons and the collecting duct will again be lined with that transport epithelia, and the collecting duct itself will have the capacity to further affect the flow of solutes and water out of the kidney. We have two main types of nephrons that we need to concern ourselves with. The first of these is going to be cortical nephrons. Cortical nephrons, as the name suggests, is going to be nephrons that exist only in the renal cortex, only in the outermost portion of the kidney. The second main type of nephron we need to concern ourselves with is going to be referred to as the juxtamedullary nephron, which is a fabulously fun word to say. Go ahead and say it. Juxtamedullary. There we go. Juxtamedullary nephrons are going to have an extended loop of Henle, which extends deep into the renal medulla. Hence the name juxtamedullary means next to the medulla, with the loop of Henle extending into said medulla. This medulla is going to be a salty place, as we'll be getting to shortly, and it allows mammals to produce a highly concentrated form of urine. The blood flowing to this structure, the nephron, is going to be regulated by two main sets of arterioles. The first of these arterioles is going to be referred to as the afferent arteriole. The afferent arteriole is going to be a branch of the renal artery that supplies the nephron with blood. This includes small branches which form capillaries in the glomerulus, which allows filtration to happen. Once the blood leaves those capillaries, it enters what is referred to as the efferent arteriole. The efferent arteriole is going to be a series of blood vessels that drain the nephron itself, that pick up the materials that are retained by that nephron. This includes a group of what we refer to as peritubular capillaries. These peritubular capillaries are going to surround the proximal and distal tubules and will collect materials that are retained by those proximal and distal tubules. Finally, there's going to be the vasa recta. The vasa recta is going to be a series of capillaries that serve the loop of Henle and play a special role in maintaining salts, especially the high salt concentrations in the medulla of the kidney. The nephron is responsible for processing the filtrate coming out of the blood vessels. The first step is the filtration step. That is going to be going on in Bowman's capsule. Bowman's capsule is going to be an extended segment of the nephron where the blood that's been filtered is collected. In this case, blood pressure from the afferent arteriole will force blood plasma through a series of very loosely connected epithelial cells. That blood will be strained out so that the red blood cells and the large proteins are maintained in solution, but the solutes and the water are allowed to pass between those cells into the glomerulus. Blood pressure forces fluid from the blood in the glomerulus into Bowman's capsule. In this case, filtration is going to be non-selective for different types of solutes, with the exception of large proteins and blood cells. In this case, Bowman's capsule will allow water and salts to move into Bowman's capsule and then into the proximal tubule. The proximal tubule is going to be the primary place where sodium and water is resorbed within your kidney. Up to about 70% of the fluid that flows in through the glomerulus is resorbed in the proximal tubule. This is being driven by the resorption of sodium and chloride. This zone is also going to be responsible for the maintenance of body pH and will also be the location where specific toxins are being secreted from the blood vessels directly into the filtrate. The main processes going on here are, first, the activity of sodium-potassium pumps. Those sodium-potassium pumps draw down the amount of sodium inside those cells. Those cells then have channel proteins which allow sodium to flow in, allowing sodium to be moved into the blood. Also, through the use of things like co-transporters, such as a sodium glucose transporter, things like glucose and amino acids can be transported from the filtrate into the blood. The next main region of the nephron is the descending loop of Henle. The descending loop of Henle is going to be responsible for the resorption of water. Aquaporins, or proteins that allow for the movement of water across cell membranes, are going to be heavily present in this zone. These aquaporin proteins make those transport epithelium very permeable to water. There are no channels for salts or solutes, however, in this zone. As a result, this is going to be a zone where you only have aquaporins and water moves from the filtrate to the blood because the blood and the interstitial fluid are going to be saltier. The ascending loop of Henle is going to be responsible for the resorption of sodium chloride. This resorption of sodium chloride is going to be due to the processes going on again with those transport epithelium, specifically through the use of ion channels that will allow salts to move across from one side of the cell to the other, but will not allow water to move across. This is going to occur in two main zones. The first zone of the ascending loop of Henle is referred to as the thin segment. This thin segment will allow sodium to diffuse from the filtrate into the blood via passive facilitated diffusion. Once you get up to the thick segment, though, you reach an area where you start to see increasing amounts of active transport of that sodium and chloride from the filtrate into the blood. As a result of these steps, the filtrate becomes progressively more dilute in terms of solutes. Having moved through the loop of Henle, we next come to the distal convoluted tubule. The distal tubule will be responsible for the regulation of potassium in the body. It will be responsible for the resorption of some amount of sodium and chloride, and will be responsible for the regulation of pH. In this case, potassium and hydrogen ions are secreted from the body solution into the filtrate. At the same time, sodium and chloride and bicarbonate ions are then absorbed from the filtrate back into the body. Within the juxtamedullary nephron, the osmolarity gradient created by the pumping of sodium ions at the thick rising limb of the loop of Henle sets up what we refer to as the countercurrent multiplier mechanism. And this mechanism will drive the absorption of salts and water in that juxtamedullary nephron, specifically and with the loop of Henle. This absorption of water and salts by the loop of Henle will be critical for the mammal's ability to maintain its osmolarity. But that is the topic of the next lecture. And so until then, stay safe, stay healthy, and I'll see you later. Okay. Coming on